Topic for presentation is prospects of 3D cell culture analysis and its implications in 3D bioprinting. So basically, I'm from NIT Calicut. So we are situated in the foothills of Western Ghat with a very beautiful picturesque landscape. It's a very green, and we have aimed at student strength. And around 15 departments have 11 undergraduate postgraduate programs, PhD alone. To the Department of Bio, and we are number 25, 2024. Number 2, number 8. So
sorry, still your voice is breaking.
It is still breaking, sir.
Yeah, it's still breaking.
Let me see, sir. Actually, my signal strength is also good. Not an issue with my signal strength. It is good.
Okay, you can start. You can start. If there is anything, I'll let you know, sir. Okay.
Just a moment, sir. Is it okay? Is it okay now?
Yeah. Now it's okay, sir. Can carry on.
Okay. Presentation. I have two different parts. We'll focus on something called as cell culture technologies. And in the second part, we'll talk about the implications of these cell culture technologies in 3D bioprinting. And if there is any issue or any questions in between, please do stop me. I'll be happy to answer you. Or if you're not able to hear me properly also, please notify me. Okay. So before first we have to culture. So basically, I'll have the laser point here. What is cell culture? Cell culture is basically something you call it as in vitro cell culture. Why it is called in vitro? Because the in vitro, the term comes from Latin. In vitro means inside a glass. So it is basically you're culturing inside a glass in a laboratory technique that involves growing of cells from a particular organism in controlled environment. This is very important. And you do this basically for research and diagnostic purpose. But how do you do that? So the cells are basically removed from the desired organism. It can be either a plant cell or an animal cell. So if you're working on animals, you call them as animal cell culture. And if you're working on plants, you call them as plant cell culture. So today we'll focus only on animal cell culture. Okay. We're not going to focus on plant cell. So when you take out these cells in a controlled nutrients, hormones, and to mimic the environment body, you mimic the lab. Have a specific set of parameters, includes a pH and your osmotic pressure. You have to do all of these things. It is purely for research purpose, trying to understand cell biology, disease mechanism, and drug toxicity. And in terms of diagnostics, you can do this to identify pathogens, that is basically your viruses or any other bacterial infections that can cause diseases. And in terms of biopharmaceutical industries, you culture them to produce enzymes, hormones, and vaccines. And also you have something called as fascinating stem cells, which you use them for stem cell therapy. Cells, the basic difference between your plant is that the animal, whereas your plant cells, so this cell wall is pure, and in your plant, which is occupying most of, and they are produce their own food, syndrome, lysosomes. These are all absent lysosomes, which is basically responsible for breaking down your. So the cells are the basic building blocks of all living things. And in your human body, there are trillions of cells. And what is the purpose of these cells? These cells provide the structure. They take in convert food into nutrients and carry out all the metabolic activities. So cell contains the body's hereditary information, that is basically the gene. Inside the nucleus, you have your DNA. So what are the sources for the cells? So cell sources can be of three different types. It can be either autogenic, allogenic, or xenogenic. What is autogenic? When you take the cells from the own body, you call it as autogenic. Allogenic is when you take the cells from the same species, it is called allogenic. Xenogenic is when you take the cells from a different species, that is called xenogenic. And this is the level of organization of tissues in your body. So basically you have the cells, these group of cells will make up the tissue, and these tissues will make up the ECM. You call them as ECM, extracellular matrix. And this ECM builds the organ, and organs together build up the organ system, and these organ system determines an organism. So this tissue level hierarchy is very, very important because you start with the cells. You build the tissues will build the organs, and the organs will build your organ system. So the basic thing is that any problem that you have in an organ, nowadays we focus on rectifying what is a problem at the cellular level. That is why cell therapy is more important. So one simple example of cell therapy is your stem cell therapy. So any problem that you have, you focus on stem cell therapy, which means that you rectify the problem at the cellular level. So when the problem is rectified at the cellular level, the tissues are well formed. When the tissues are well formed, the ECM is well built. When the ECM is well built, the organ is properly functioning. So that is why any issues in your body, you focus on the cell. If the cells are pretty okay, then everything is perfectly all right. So where the different will be cell proliferation. What is this and differentiation? Fundamental processes that are cell development. Cell addition is basically material, able to attach to a ECM for the cell survival, proliferation, migration, and differentiation. And this is also very, very important when the cells communicate with each other and they elicit a particular biological response. Cell proliferation is nothing but cell division, which is basically increase in differentiation is a process. As specialized cells, they have to attain a function in this particular lineage. Basic diagram. So what you see here is a that is getting attached to the substrate. This is the front part of the cell, and this is your substrate. And at the center, there is your actin cortex. So when there is a tension, what happens is cell has to move in this direction. So when there is a tension, what happens? This actin, basically the microtubules, the actin this particular end, and this will push or protrude your lamellipodia. So that is why this part is called as your protrusion. And when this there is one more protein which acts behind the cell, which particular portion. The protrusion happens, and these controlled by group of proteins. So the contraction is controlled by a myosin, and the protrusion actin. So when happening, cells will start moving direction, unidirectional way. The cells are
Sorry, Gopal Shankar. It's the voice is breaking.
do one thing. Sir.
I I can't hear you.
Sorry.
இப்போ கேட்கல கேட்கல. I can't hear you. So now speak, sir.
Sir, is it no?
Now is it okay? Like I can't hear you. Can you speak?
Voice is very putting the max. It is at 100% now, the volume.
Okay. Now it's audible. Now it's audible. Okay. Carry on.
It is.
Yeah. Now still there is a break in the voice. That's it. The presentations are perfect, but like the voice is breaking.
Just quit the. Is it okay?
Uh, uh, is it okay
There
for
is
others?
too much of disturbance, or if not, I can continue.
So it's too much of disturbance. That's it. Like yeah. So there's a gap between one sentence and another.
Okay,
Yeah.
then it's better that I quit and come back again.
Yeah. That'll be better.
Okay.
So the audience, please message if the voice is breaking. The voice is breaking for me. Some says the voice is clear for us. Gopalakrishnan says the voice is breaking. So if anyone has problem in voice break, you can just message so that we can get to know. Okay. Thank you.
So can you hear me now?
Yeah, it's audible. It's audible. Can you just present?
It's audible. Okay. Okay. So I think I stopped here on this particular page, right?
Yes.
Yes. Okay. So now it's audible, right? Without any
Yeah.
disturbance.
Now it's audible. Now it's audible. Now it's audible.
Okay. So we were talking about cell addition, which is basically the cells have to adhere to the extracellular matrix or to any substrate to enhance the functions of cell survival, proliferation, and just a moment. Proliferation and migration and differentiation, which plays a vital role in cell communication and regulation. And cell proliferation is basically to increase in the number of cells. And cell differentiation is a process where the cells become specialized and they attain a particular function. Because all undifferentiated cells do not have any functions. When they attain a function, they become differentiated. And differentiation is a multi-stage process which will give you some specialized functions to the cell. And this is the basic cellular attachment process where we talked about the lamellar podium, which is controlled by one protein that is called as your actin filaments, which allows the cells to move in a forward direction. And the backward contraction is initiated by this myosin, which is another protein. And by this mean the cell moves from one particular location to another location in a unidirectional motion, and the cells are attached to the substrate by means of integrins that is called as your focal contacts. So all your undifferentiated cells are called as your stem cells. So any cells which are undifferentiated, which means they do not have any specialized function, you call them as stem cells. So the differentiated cells have a specific functions, whereas the undifferentiated cells do not have a functions. So the differentiated cells can perform a function in the body, while the undifferentiated cells will help in replenishing your old or injured dead cells. In terms of proliferation, the differentiated cells do not undergo proliferation, while your undifferentiated cells, the stem cells that are responsible for your self-renewal process. And in terms of development, what happens is that the differentiated cells are results of cell differentiation process, and they generally give rise to structures and functions. And undifferentiated cells, also known as stem cells, and they have a potential to develop into any different types of cells. So you see, this is a stem cell. A stem cell can become a fat cell, muscle cell, sex cell, human cell, epithelial cells, or whatever it is. That is why it is undifferentiated. So when you talk about stem cells are cells that can develop into different types of cells in your body, and they can renew themselves over a period of time. So when you talk about the stem cells in terms of potency, you can classify them as totipotent, pluripotent, multipotent, oligopotent, and unipotent. So what is the difference between all of these different classification of stem cells is that totipotent is purely embryonic stem cells, which is mean that you have it in the zygote. So what happens is that you see this is a fertilization of a egg and sperm. And first you develop a 2-cell stage, then the 4-cell stage, 8-cell stage, then the 16-cell stage. And at the last, you develop this blastocyst. And the cells, what you see here is called as your inner mast cells. So the totipotent cells is basically embryonic stem cells, which means that the cells that you isolate within this fertilized egg till the 16-cell stage. So again, it is during the first 16 weeks of this fetus development. I'm sorry, the first 4 weeks of this fetus development. And that is why you call them as extra embryonic stem cells. Pluripotent stem cells is a stem cell that you isolate from the inner mast cell. This is your blastocyst, and the cells that you see here, these are called as your inner mast cells. So this is called as your pluripotent stem cells. And all this multipotent, oligopotent, and unipotent are taken from the adult stem cells. So a typical example is your hematopoietic stem cells, your lymphoid or myeloid stem cells. Here is the satellite stem cells or epidermal stem cells. So what is the difference is that these totipotent and pluripotent, they can differentiate into any type of cells that you want. So they can become a sex cell, they can become a somatic cell, they can become fat cell or a bone cells or whatever it is. When they become an adult stem cells, their differentiation is particularly limited in one particular direction. And this is an irreversible process. Once they become a progenitor cells, precursor cells, they have to become a progenitor cells, and they have to follow this pathway. So this is not an irreversible process. So they keep on moving in this particular lineage. Okay, so the basic difference between the totipotent and pluripotent is that the totipotent stem cells are basically the extra embryonic stem cells, which is taken from the first few days of the fertilized egg. And again, the embryonic stem cells and your pluripotent stem cells do have ethical constraints because you have to kill the embryo to isolate these cells. So instead of this, one technology was proposed that is called as your induced pluripotent stem cells, which means that the somatic cells can be transformed into a pluripotent stem cells by means of 4 transcription factors. And this is called as your Yamanaka factors, which was discovered by the Japanese scientist, and he was awarded the Nobel Prize, which is basically converting a normal adult somatic cells to behave like a pluripotent stem cells. So now with all the basics of the cells, now let's move to the cell culture laboratory. So to successfully run a cell culture laboratory, you need all of these essential equipments as well as your consumables and instruments. So one basic thing is the incubator that is very much required. Then you need an inverted microscope, then refrigerator, then liquid nitrogen, centrifuge to separate your cells. And to work in a sterile condition, you need a laminar head, laminar flow. Then how do you culture the cells? You need the vessels, cell culture vessels, consumables, pipettes, hemocytometers basically used to count the cells, and you have a water bath. So these are the basic instruments that are required to run a successful cell culture laboratory. And what are the conditions that you need? You need a condition that is called as your pH. So what is this pH? pH is basically that the blood pH that is normally used is 7.7 to 7.4, or you call them as a physiological pH. Then carbon dioxide, we need carbon dioxide around 5 to 10%. That is basically supplied by the cylinder in this particular incubator. Then you need temperature. We need a temperature around 37 degrees. That is basically your human body temperature. And there is one more thing that is also called as your osmolality, which is basically 280 to 300 osmoles per kilogram, which is basically trying to measure the concentration of dissolved particles in the particular media. And this is very important for maintaining the osmotic pressure. So when you talk about this osmosis or osmotic pressure, we know that we should have an isotonic solution, which means that the net movement of water inside and outside the cell should be equilibrated or balanced. If there is too much of salt concentration inside the cells, what will happen? The water will flush out from the cells and the cells will start to shrink, or you call them as crenation. The opposite, when there is low salt concentration in the cell, what will happen? The water will start to move inside the cells, and the cell will start to swell, and the cell will lyse. So to maintain this isotonic solution, you need to maintain your osmolality, which is basically a key factor in maintaining the water balance and your osmotic pressure. So when you talk about the media, you have 2 different types of media. One is something called as artificial media and natural media and artificial media. What is this natural media? All the fluids that you find in your body are called as your natural media. So what is the natural fluid that you find in your body? One predominant fluid is your blood. So blood is a natural media. So that is why the tissue extracts, fluids and clots, a natural media. A chick embryo, natural media. Bone marrow concentrate, which you take from the iliac crest of your bone, natural media. Amniotic fluid, lymph fluid, etc. So these are all the natural media where the cells are growing and they're doing their vital functions to maintain the metabolic activities. On the other hand, when you want to do in vitro studies, we have to mimic the composition of all these natural medias to produce something called as your synthetic media or synthetically artificial media. So this synthetic media contains one important component that is called as your serum. And this serum comes from your fetal bovine serum, which means the fetus of the bovine. You take the blood and you separate your serum. Okay. So why do you need the serum? Because the serum contains all the different components that are necessary for the cell attachment. So what does it contain? It contains growth factors that are essential for cell attachment, lipids, hormones, and they also contain nutrients and energy sources, that is vitamins and sugars. There are electrolytes, carriers, enzymes, and there are essential factors that maintain the pH, and there are also some binding and transfer proteins that are controlling your cell addition factor. So without a serum, it is very, very difficult to maintain the cell culture media. So serum is an essential component. So this is a basic buffering system. So you know that the buffering system in your body is maintained by something called as a carbonic acid, which is basically an acid, a weak acid. So when there is an excess of ions, when there is a need of ions, it will donate H plus ions and it will convert into your bicarbonate or hydrogen bicarbonate. And similarly, on the opposite side, when carbon dioxide, superficial carbon dioxide reacts with water, it will also produce your carbonic acid. So this part of the function takes place in the lungs during your decrease in respiratory rate and increase in respiratory rate. This takes place during your, this takes place in your kidney, that is maintaining your metabolic activity. So this is the normal process buffering system that is happening in your body. So to maintain this buffering system in your cells, you need a buffer. A buffer is basically a chemical that will resist changes in your pH. Okay. So here in your media, usually you have these 2 buffers, either it is sodium bicarbonate or Herbst buffer. So what will happen is that you see here, Most of the time, your cell culture media is red in color. This is a pH indicator which is provided by phenol red. So when the media is going on a acidic scale, the color of the media will change. It will become more yellow, which means that the cells are starting to starve and you have to replace the media. And if it is more pink in color, it shows that it is going on an alkaline condition where there is too much of carbon dioxide supply. So basically, this phenol red is an indicator which will tell you that the pH is maintained at 7.4. If it is going on the negative, if it is going on the acidic side, you have to change the media, which shows that there is too much of growth or there is some kind of contamination. If it is on the alkaline side, it becomes more pink, which is basically you have to check your carbon dioxide condition. So when you talk about the normal morphology of your cells, most of the cells in your human body, we know that multiple types of cells are there. Most of them, they exhibit three different characteristic morphology. So is everything okay? Is my voice audible? All good?
Yes, Gopal Shankar. It's good,
Yes.
good.
Okay. Okay. So the morphology of cells in culture, basically, it is fibroblastic, which is bipolar. You see the cells having bipolar or elongated, or you can also call it as multipolar, and they grow on the substrate. Sometimes they also have this epithelial-like cell morphology, which means they are polygonal in shape and they grow more in regular dimensions and they attach to the substrate. And the last one is something called the lymphoblastic cells, which are basically spherical in shape, and they usually grow on suspensions, which means that they do not attach to the substrate. So these two attach to the substrate, they are called as anchorage dependent cells. Whereas this one, they grow on suspensions. That is, they float on the fluids. They are called anchorage independent cells. Okay. So you isolate the tissue. After isolating the tissue, you will have to disrupt the cells. So how do you disrupt the cells from the tissue? You can go for physical agitation, that is basically by syringing or vigorous pipetting or sieving, or you can go for enzymatic digestion, or you can do chelating agents. One chelating agent is basically EDTA. What is the function of EDTA? EDTA is a chelating agent which will bind with calcium ions. So the one typical example I'll tell you is that all your vials that you use for collecting blood are coated with EDTA. The reason is that EDTA is basically a chelating agent. It will go and bind with your metallic groups. So it will go and bind with calcium. So calcium is an important component in your blood clotting. So if it is binding with the calcium, the role of calcium in two pathways, the intrinsic and the extrinsic pathway, is blocked. So your blood does not clot. And you take the samples for your analysis. So that is why we use EDT coated vials for collecting your bloods, which will prevent the clotting of blood. And you take your samples, and when you centrifuge your blood samples, you get two different portions. The upper part is your plasma, and the lower part you have all your RBCs. So in terms of enzymatic digestion, you have trypsin or collagenase, which will basically break down all the solid substrates in your organ. So one typical example is collagenase, because most of our body tissues are made up of protein called collagen, and this collagenase will break down your collagen and it will release all your cells. If you go for trypsin, the same concept, but here you have two types. One is your cold trypsin, another one is your warm trypsin. So when you talk about animal cell culture, again, there are two different types. One is something called as your primary cells, and the second one is something called as your secondary cells. And in the secondary cells, you have three different combinations. One is in-fight cell culture, continuous cell culture, and transform cell culture. So important thing in cell culture is something called as your subculture. So what is the subculture? Subculture is basically a portion of cells is transformed to a new vessel with the fresh growth media, which will provide more space and nutrients for the cells to grow. So there is no death of cells because of lack of nutrients or lack of space. So when do you subculture is when you reach this 90% of confluency. So maybe this is day one, you see there is only 10% of cells that are living on the plate. Probably this is on day two, you see there are 30%. They're getting crowded and spreading the plate. And day three, there is 50%. And once you reach this particular concentrate, this particular range of 90% of cells that are completely plated on your particular vessel, this is called 90% subculture. And you take this point where you understand that the cells need to be refreshed with new media and they cannot grow more sufficiently here. So they have to go to a new media. So subculture is a particular point where you take a particular portion or of this particular cells and you put them in a fresh media. So why do you have to do subculture? Subculture is basically to increase the number of cells in the culture, and you have to enhance the lifespan of your cells, and you can prevent the cells from dying and ensure the propagation of cell line, and you produce more homogeneous cell line. So this is the basic growth curve. You see here, this is the total cell yield, and this is your timeline. And this is the point where it's seeded. And you see immediately after the first phase, this is basically the log phase. You see that you go for subculture that is called primary subculture. And when you, after this first subculture, again, you go for the second subculture. And you see that the growth curve goes on. And at this point, the cells have to be in the secondary culture. You call them as cell line. I'll talk about that later. And if they are still going on, that is called continuous cell line, that is transformed. If not, the cells will die. That is called senescence and aging. So what is senescence? Senescence is that the cells have lost the ability to divide. Though they are metabolic active, they have lost the ability to divide. And senescence and apoptosis should not be confused. Apoptosis is something called as your programmed cell death. Senescence is something called as your cell aging. Now, we'll talk about something called as cryopreservation. So what is this cryopreservation? So when you have too much of cells, you don't want to waste them. Instead, you can store them. So how do you store them? You can store them by using some kind of cryoprotectant that is called DMSO or glycerol. Most of the time we use DMSO. Okay, DMSO is dimethyl sulfoxide, and you store them at temperature that is around minus 130 degrees. So what is this function of DMSO? How is this DMSO being used? And you store them in the cryoprotectant and put them in minus 183. And to revive them back, again, you have to thaw them in a warm temperature and provide them adequate medium and serum for them to revive. So basically, the concept of cryoprotectant is that it will protect the biological tissue from freeze damaging. So what happens in freeze damaging? You will form the ice crystal formation. So when these ice crystals are forming, these ice crystals can basically kill your cells. So cryoprotectants, what they do is that when you add this DMSO along with your cells, this DMSO will go and replace your water molecules in the cells. So when this DMSO is being replaced, the prevention of ice formation and cell death is being completely hindered. And they have lower melting point than water, which protects the cells. That is an important point. And they also do not allow the translocation of components through the damaged membrane at the time of repair. So one typical example is DMSO, most commonly used. So there are two ways you can do that. One is either by slow cooling or by a rapid cooling. So for rapid cooling, you need liquid nitrogen. The temperature of liquid nitrogen is minus 196 degrees Celsius. Whereas in slow cooling, what happens? You slowly cool it and bring it down to a temperature of 130 degrees Celsius. Okay. But here in vitrification process, it is minus 198 degrees Celsius. So you need liquid nitrogen. So what is the concept here is that you see here, 0 is the freezing point. When you go down in temperature at minus 40, what happens? The ice nucleation temperature. So there is an ice phase formation. But when you go below minus 120, you can bypass this ice phase, that is a liquid phase, and directly go to the glass phase. So when you go to this glass phase, there are no crystal formation. When there is no crystal formation, your cell is not dying. So the concept is simply that you take the cell and rapidly quench in minus 198 degrees Celsius, that is liquid nitrogen. You will bypass all of these phases. There is no freezing point, there is no ice nucleation temperature, and there is no ice phase, which means that you directly bypass the liquid phase and go to a glass phase. So when you have this glass phase, there is no crystals. So this is the role of cryoprotectant. So when we talk about cell culturing, two terms are very important. One is 2D cell culture and one is 3D cell culture. So what is 2D and 3D? So 3D is that 3D is basically that you have three dimensions. Basically, your eye can see in three dimension because you see the length, the width, length, and the depth at the same point, three dimensions, three axes. So 2D cell culture is basically the cells which are grown on a flat surface. You grow them on a flat surface, and the cells are flat and do not represent your real cell physiological morphology. All the cells in your body are growing in three-dimensional. But when you do it in 2D culture, they are not in three dimensions. They are interacting with only one particular phase. Other two phases are left exposed. So 3D culture is the best place, that is a 3D space that is more essential for cell growth. And when you do that, the cells will form aggregates, or you call them as preoids, and they have more space for their interactions. So here you see the cell-to-cell contact interaction happens, the cell-to-scaffold interaction happens, and the cell-to-media interaction happens. So in 2D, the cells will interact only with the culture media and the petri dish, and they will only form a monolayer, which means a single layer of cells. You cannot grow more than that. And this is a simple process, inexpensive, reproducible, but most of the time it cannot mimic the natural conditions in your body. In contrary, in your 3D cell culture, what will happen? The 3D cell culture will interact with the scaffold surface. They can grow in variety of cells. I mean, a variety of ways that include cell culture, that is reaggregate cell culture, hydrogels, or you can use a bioreactor. And the 3D cell culture system is more complex and it can be more biomimicking the in vivo environment. So if you want to mimic something under the lab conditions, you have to go for 3D cell culture. So how do you use 3D cell culture? Basically use a scaffold. So these scaffolds, we'll talk about them later, but you need scaffolds for this. So what is the cytotoxicity testing? So now we have the cells, you have put the cells. Now we have to understand if my cells are growing or not. So cyto is basically cell, toxicity is basically toxic or not. So cytotoxicity is basically cell toxicity. So there are a number of ways to do that. There are multiple assays based upon staining techniques, microscopic techniques, or absorbance based techniques. There are multiple techniques. So basic term, as a name indicates, cyto is basically cell, toxicity is toxin. So cell toxicity. So how do you do that? We cannot simply do that. There are some standards in toxicity testing. So we follow the guidelines that is provided by the International Organization of Standard, that is called the ISO. So under ISO, there are multiple categories. So we focus on this particular category called as ISO 10993 series. So this series will talk about biocompatibility testing and assessment. So what does it does? It is basically a combination of written evaluations or biological endpoints that will evaluate the compatibility of a particular device. And this is controlled by FDA, which is basically your Food and Drug Administration located in US. So when you talk about this biological endpoint evaluations, there are a number of studies. So you see here, these are the biological endpoints. And on the left-hand side, you have something called as categories. This is a medical surface device, externally communicating device, and implants. So under this implant, you have a bone or a tissue implant, blood interacting implants, circulating blood in terms of external communicating, tissue, bone, and dentin, blood path, and indirect. In terms of surface, intact skin, mucosal membrane, and you have the breached or the compromised surfaces. And these are all ABCs are the classification of risk, whereas A comes under low risk classification, B comes under moderate risk classification, C comes under moderate high risk, and D comes under high risk. Okay. So, and again, here you have time durations where A is basically below 24 hours. You put the material within 24 hours and you remove it back. And B is something between 24 hours to 30 days. And C is basically above 30 days. So here we are focusing only on this particular point that is called cytotoxicity, which is basically trying to understand if this material will allow the cells to grow without any toxic response. That is a simple thing. And you see here, for all of these devices, you have this E, which means that this is mandatory. We have to study this cytotoxicity. You cannot omit them. Okay. So what is the cytotoxicity classification of cytotoxicity? I've told you cytotoxicity is nothing but cell toxicity, which will measure the parameters that are associated with a cell death and analyze the toxic response. So if it is toxic, what will happen? It will rupture the membrane and the cells will start to die. If it is not toxic, the cells will be alive. So when you want to study cytotoxicity, how do you study them? You understand if they are viable or not. Viability is basically measuring the health conditions. So you can do them either indirectly or directly. And if they are viable, what they have to do? They have to multiply. They have to grow or they have to increase in number. So basically, the cell viability is defined as the number of healthy cells in a sample. And proliferation of the cells is the vital point or an indicator for understanding the mechanism of action of certain genes, proteins, or pathways that are involved in your cell survival or death after exposing to a particular toxic components. Let's say that I have an anti-cancerous drug. I have an anti-cancer cell line with me. So when I put this anti-cancerous drug with the cell line, obviously what will have to do? These cell lines have to die. So the dosage of this anti-cancer drug will determine the death of this particular cells, because again, dosage we have to fix. So this is basically the cytotoxicity, where you try to understand the viability or proliferation. Now let's take a normal cell. You take the normal cell and you treat it with the same drug. The cell should not die because it is not a non-cancerous. It is a non-cancerous cell. Only the cancer cells should die. Or you take any material and you interact with the normal cells, the cell should survive. There should be an increase in proliferation, which directly indicates the viability of your cells. So again, cytotoxicity is one of the biological evaluation, or you call them as a screening test that uses the tissue cells under in vitro conditions to observe growth, reproduction, and morphological effects of medical devices. So when you talk about this viability, what you exactly try to understand is basically the metabolic activity of the cell membrane integrity, whether it is broken or damaged, and the morphological evaluation of your cell. So the first thing we'll talk about what are the different types of cytotoxicity studies. Before that, let's talk about the cell sources that are available for in vitro studies. So the cell sources can be from number of sources. So if it is from the donor, you can take it from a human or animal. If it is from a tissue, you can take any tissue and you can extract the sources. And based upon the divisions, the primary cells have a limited divisions, and continuous cells have an unlimited division. We call them a cell line. We'll talk about that later. Or you can take them from tumor tissues, or you can also take them from immortalized cells. And when you talk about cells, one is adherent cells, other one is non-adherent cells. And this concept is very, very important. That is something called as your TERT, that is human telomerase reverse transcriptase process. I'll talk about this bit later. Okay, no, not later. Now it's sorry. So you see here, how do you make the cells immortal? Immortal is basically nothing but the cells can keep on dividing continuously without without attaining the stage of cell senescence. So how does it happen? You see, this is the basic chromosome. Okay, this is a chromosome of your cell, and at the end of the chromosome there is something called telomeres. Okay, so as the cell divides, the length of the telomeres will shorten. So when the telomeres are completely lost, the cells will stop dividing. And at this point, the cells will enter the stage of senescence. And this point or this limit is something called as your Hayflick limit. This is very, very important. So the idea now is that if I'm able to break this telomere shortening, if I'm able to allow the telomeres not to shorten, then I can make my cells continuously divide. So how do I do that? I do that by by introducing an enzyme that is called telomerase, which will keep on adding new telomeres to this end. So this is basically done to impart immortality in your cells. So that is something called as your forced expression of telomerase reverse transcriptase enzyme. So when this enzyme is added here, what will happen? The cells will keep on dividing. So that is the first option. The second one, you can also go for activating your oncogenes, that is basically a cancer causing genes, and cancer cells have the ability because they can keep on dividing. And the third one is that you can also, there is something called as p53 genes, that is nothing but the tumor suppressing genes. If you are able to suppress this, then you can also keep your cells in immortal condition. So these are the different ways where you can make a normal primary cell into a secondary cell line. So one common method is basically your telomerase reverse transcriptase activation process. So this is the simple structure of cell types and structure: differentiated cells, stem cells, monolayer, and suspension. We saw them. Now let's go to cell counting. So what is the cell counting? So now you have the cells with you. Now you have to count the cells. So how do you count the cells? I need something called as your hemocytometer. And with the hemocytometer, how do I identify my cells are healthy or not? Then I have a dye called as your trifine blue, which is basically an azo dye. And this dye is impermeable with your live cells, and it can only enter the cells which are being broken. So when they enter the cells which are broken, they bind with something called the intracellular protein, and they give a blue color. Okay, so you see here, this is the basic hemocytometer. You have two chambers, chamber 1 and 2, and you take the cells and you mix it with this dye. And after mixing it with this dye, you can understand the viability of the cells, viable cell count, non-viable cells, and also you can understand the percentage viability. And this is a basic hemocytometer. This is how it looks like. You have 9 squares. Most of the time we count the squares in the corner and the center. So 1, 2, 3, 4, and 5. We count the squares, the number of cells counted. So let's say that I have 50 cells, 50 divided by number of squares into dilution factor. Dilution factor is nothing but how much cells you take and how much volume of your dye you take, and you divide by that, you get the dilution factor. And this 10,000 is basically that one square is representing 1 by 10th of a ml. So that is why in order to make it into ml, we multiply by 10,000. That is a factor which comes along with the formula. And this is how it looks like. So when you stain it and view under the microscope, you see here, these are all non-viable cells because the dye has penetrated inside the cells and it becomes stained. And these are all viable cells. So again, this is a non-viable cell, viable cell. So this is how it looks like. You can do it manually, but now we have sophisticated instruments that is basically called your automated cell counters that will count and give you the cell number. So I've counted the cell number. Now I have allowed my material to interact with a particular drug or a component. Now I have to understand the viability. So one simple experiment that is used to understand the cell viability is called as your MTT assay, which is basically based upon the MTT dye, which converts the formosyn crystal. It happens only in the living cells because it is determining the mitochondrial activity, which determines the mitochondrial activity. So this MTT, when it reacts to the live cells in the mitochondria, there is something called the mitochondrial reductase that will produce a formosyn crystals that is basically purple in color. So you see the chemistry is like that. It is a rapid calorimetric test where you see that in presence of NADH, it gets oxidized, it becomes NAD plus. When this oxidation process in presence of this mitochondrial reductase enzyme, you end up in forming this formosyn, which is basically a blue color. And also you can also identify the half maximal inhibitory concentration that is called the IC value. That is basically the measure of potency of the substance in inhibiting a specific biological or a biochemical function. Basically, you see here. So this is the cell viability and this is the drug. You see at this 50%, the IC50 value is somewhere here. That is basically 25.2. So it is nothing but the amount of drug that is needed to inhibit a biological process by 50% under in vitro condition. That is called as your IC50 value, half minimal inhibitory concentration. So you can also do something called as your live and dead assay that is basically based upon fluorescent tagging of molecules. So one common live and dead assay is your AppCam kit. So basically what happens, you have the live cells that are green in color, then you have your dead cells that are red in color. So for this is how it looks like. You see here, these are all the live cells which are green in color and the dead cells. And most of the time you have to use a confocal microscopy because or a fluorescent microscopy, because if you use a bright field microscopy, you don't get any details or information about the cell morphology. So you have to use a confocal or a fluorescent microscopy. And this is basically the confocal laser microscopy. You see the advantage is that you can separate the cells based upon their healthy conditions, whether if they are live or dead, and you can also merge them, and you can also produce 3D stacked layers. Okay, so it is very, very essential or very, very vital in obtaining high resolution optical images with depth sensitivity and allows your protein localization in a particular specific cellular component. The reason is that with your microscope, when you focus at one point, all other points becomes blurred. So basically, if you have a material that is a 3D dimension scaffold, with a normal microscope, you cannot go below a particular point. You have restrictions or limitations. But with a confocal laser microscope, what happens is that you can go in the depth axis, that is the zeta axis, you can go down further and try to look how the cells are distributed in the scaffold material. And also you can also use a flow cytometry, which will sort your cells based upon the fluorescent tagging. So the basic principle is that you pass the cells through a single file, and this is basically a mixture of cells, and you tag them with some fluorescent molecules. So when they interact with laser, there is something called excitation wavelength and emission wavelength. So they get excited at a particular wavelength, go to the higher stage, and when they come back to the ground state, they give some kind of emission that will give you a different wavelength, that is color. So based upon this fluorescent detectors, you can quantify the cells, that is cell sorting. And this is how the data looks like. So let's see this is the viable cells and the necrotic cells, and you can also identify the cells which are in early apoptosis and late apoptosis, and you can get a graph. And morphological analysis, one typical example you can go for scanning electron microscopy, that is SEM. And you see here, this is the normal morphology of A431 cells, and this is MG63 cells and ACAT cells. But when you give a drug, you see that in the cancer cells, these are two cancer cells and this is the normal cells. When you give the drug, you see that the cell structure is getting damaged or it's getting broken. But here in the normal cells, there is no damage. They typically look the same cluster formation and the agglomeration is available here. But you see here, they're getting damaged and broken. And here you can see that the group of cells are getting attached to the pores of your scaffold. You see, these are the cells that are getting attached to the pores. And here you can see the extension of the cells. That is basically the lamellar podium, which we talked in the beginning of this in the slides. We talked about this lamellar podium. You can see them; they are extending. And also you can also use transmission electron microscopy to understand if there is a cell rupture or not. Again, you see here, this is a cancer cell. A431, and this is MG63 cells. As you increase in a drug concentration, which is basically anti-cancer component, you see that the cells are bulbing and their nucleic contents are getting out. But here, this is a normal cell with a normal nucleus. Okay. And there is also something called gene expression analysis. What is this gene expression analysis? We know that all the multicellular organisms express a particular set of genes that contain the same amount of DNA. And these particular set of genes will determine the protein function or the functional RNA that gives a unique property. So in all the eukaryotic systems, especially in humans, gene expressions involves a multi-step and gene regulation that occurs at multiple stages. And these genes are regulated at the transcription level. For example, you see here, this B cell, the B cell to produce a macrophages or this fibroblast to become more pluripotent stem cells, these four factors are essential. Similarly, the pancreatic exocrine to develop into islet cell, these three factors are essential. So there are specific set of genes that are very unique for these cells. And when you quantify these genes, whether these genes are upregulated or downregulated, you can understand if the cell function is properly being maintained or not. So to do this, you obviously need an RT-PCR. So RT-PCR you do, and you get the complete set of genes. And this regulation of gene expression is highly important during the development of an organism. And these set of regulatory proteins will turn on or turn off particular genes at the right time of organism that is essential for the normal development of organs and organ system. And finally, we also we also know that there is something called histology. Histology is basically a microscopic anatomy, or you call it as a microanatomy. It is a branch of biology which studies the microscopic anatomy of biological tissues. And when we talk about the animal tissues, broadly classify them as muscle tissue, nerve tissue, or connective tissue. So how do you do this? You take a piece of tissue, you fix them, dehydrate them, then you go for embedding. So then you have a microtome and you cut them. Then you fix them. There are multiple types of stainings. So one typical staining is that is called as your H&E staining, hematoxylin and neosin staining, which will give the cytoplasm and the cell content. Okay. So sterilization is basically how do you sterilize your materials before going for in vitro studies. So there is something called heat sterilization. There is filtration. So media, you can do filtration. The sound waves or ultrasounds are there. Moist heat sterilization, most of them autoclave. And also you can go for dry heat sterilization. And mostly in terms of tissue, we go for irradiation, that is basically using gamma rays or UV rays. And pressure is also used. And sometimes you can also go for solar disinfection process. So it depends, and it depends upon the application, what you're targeting. And it also depends upon the material that you're using. So based upon your material that you use, you can go for any of these types of sterilization process. And one important challenge that you have in cell culture is contaminations. So these contaminations are basically of two types. It can be either a chemical contamination or it can be a biological contamination. So chemical contamination can come from your chemicals that will basically harm your cells. And biological contamination it is basically your bacteria or fungi or yeast, which comes along with your personal hygiene. So most of the time, the contaminations are basically because of accidents and mistakes, that is around 33%. Cell culture media and reagents around 12%. Particles or aerosols 15%. That is basically during the handling process. Culture media additive 6%, and microorganisms that are growing independently on all your culture vessels. That is important. So sterilization, perfect sterilization is very, very important to avoid all your contaminations, and personal hygiene is also of priority when you're working in cell culture. So this is all about the part one, where we talk about cell culture technologies. Now let's move on to the next one, that is something called as your 3D printing. Okay. So just a minute.
So any doubts till here, we can just ask some doubts, or like we can just continue with 3D printing.
Yeah, I'm open for any kind of questions.
Okay, because before going for 3D printing, if there are any doubts on cell culture, so that the 3D printing will be the next part, with the cell culture will be doing the 3D printing. If there are any doubts, questions, you can post it in chat, or I'll unmute. If you are okay, then we can continue.
Okay, I think there is no response, which means that I will continue.
We can continue. I think so. Okay.
Okay, now we'll move on to something which is interesting and fascinating, which is also booming. And there is a lot of hype and hope surrounding this 3D bioprinting. So now let's talk what is this 3D printing. So before we go into 3D printing, we should understand the concept of tissue engineering and regenerative medicine. So generally call them as term TERM. What is tissue engineering? Tissue engineering is basically an interdisciplinary science that focuses on developing or regenerating damaged tissues or organs. And regenerative medicine is basically the replacement of damaged tissues by utilizing three different concepts of tissue engineering. So if you want to successfully regenerate a particular tissue, then you will have to involve this tissue engineering. So tissue engineering has three triads. One is something called as your scaffolds, that is basically your biomaterials. Then you have your bioactive factors, that is basically your signaling molecules. Then importantly, your cells. So any tissue engineering without one of these components cannot be successful. So it is essential that you have all these three components that are synchronously combined together to produce a synergistic effect. Okay. So why do you need term? There are a lot of reasons for term, because there is a question. Maybe I'll answer that question later because I need to read it. I'll answer that question later. I'll just
Yeah, yeah.
continue with the
Just
story.
continue the sessions. We'll have the
Yeah,
questions later.
we'll take up the questions later. Okay, thank you for that. So why do you need term in biomedicine? The first reason is that we need to replace structures that are getting damaged, or we need to improve or replace tissues that can be the skin, muscle, bone, whatever it is. And also sometimes you have to repair heart, kidney, etc. And ultimately when you do this, you save life. And problems what we have is that we have lack of organs, donor morbidity, and we also have graft rejection, therapy failure, and etc. So the gold standard method for tissue engineering is that we have to go for grafting. So grafting is of three different types. One is called as your autograft, which is basically you take it from your own body. Then there is something called allograft, that is basically you take it from the same species. And xenograft is when you take it from the different species. So though we have the gold standard, we still have a lot of problems, because there is a possibility of disease transmission when you go for allogenic or xenogenic source. And again, there is also therapy failures, graft rejection, etc. So to overcome all of these issues, we go for something called as your tissue engineering application. And I have told you that tissue engineering runs on the principles of something called as your biomaterials or scaffold technology. So these two terms are same, biomaterial scaffold, it's the same. So what is a biomaterial scaffold? It is basically a biological or a synthetic. It need not be only biological, it can be a synthetic also. Substance which is basically introduced into the body as a part of a medical device, implanted medical device, which is basically repairing or replacing an organ or a body function. So basically, this biomaterials will serve as a building blocks. It will serve as a template so that new tissues can grow. It will increase the bioactivity at that particular point. And when there is an increase in bioactivity, obviously there'll be an increase in regeneration. And when these biomaterials are there, they become osteoinductive, which means they will instruct the cells which are there to do some kind of physical exchange, chemical exchange, and morphological exchange of signals. And when all this is happening, the damaged site is getting reinforced, which is mean it's getting repaired. And when you do all this simultaneously with the help of these biomaterials, you can also deliver some kind of therapeutic agents that can be any kind of drug. So the regenerative medicine, again, you can classify them with scaffolds or without scaffolds. So without scaffolds, it is cell therapy, internal medicine. With scaffolds, you call them as tissue engineering surgery. And an ideal scaffold material for tissue engineering, it should be biocompatible. It should not cause any harm. It should have sufficient porosity, which is very, very essential for the cells to move up and down your material. And it should have some kind of mechanical properties that are mimicking the targeted mechanical properties of the tissue. It should be biodegradable, because biodegradation is essential that the material degrades. And when the material degrades, there is a simultaneous new tissue formation. It should be bioactive, which means that it should interact with the surrounding environment without causing any immune reactions, infection resistance, and it should be non-toxic, and ultimately it has to be cost effective. So what are the biomaterials that are available? It can be of synthetic origin. If it is of synthetic origin, all the material PCl, polycaprolactin, polylactic acid, polyglycolic acid, or their combination PLGA. If it is natural, most commonly you have collagen, because we use collagen so widely is that your body structural protein. Around 30% of the body structural protein is collagen. So any tissue you take in your body will have collagen. Or you can also go for hydrolyzed collagen, hyaluronic acid. Again, hyaluronic acid is your ECM component, or you can also use chitosan. So what are the key factors that are responsible when you go for this fabrication? The source of biomaterial, how much mechanical properties that it has, the biocompatibility of the material, and the fabrication methods. And what type of scaffolds it can develop? It can be either hydrogel. Hydrogel is nothing but the material which has high content of water. Or it can be nanofibrous, that is by electrospinning, solid porous scaffolds, but it's by lyophilization. This is very important. Decellularized scaffolds, I'll talk about that later, or biofilms. And here, these are the different fabrication technologies that are used for producing scaffolds. So the most conventional technologies, these ones, the gas forming, solvent casting, freeze drying, particle leaching. And on the other end, these are all the modern techniques: 3D printing, 4D printing, melt electrospinning, or electrodynamic jetting process. So now let's come. So as I talked about 3D printing, now let's talk about what is 3D printing. So the 3D printing is basically a machine that will print structures trying to mimic the body parts. So what is the history of this 3D printing? The first 3D printing that was discovered, or I would say invented, is in 1984. That was invented by Chunk Hull. He's basically called as the father of 3D printing. It is basically a SLA model. SLA is nothing but stereolithography. Okay, stereolithography printer. And from 1984, it got evolved. First, there was a normal printer. Then there was something called as a bioprinter, which means that you can introduce cells along with the printing process. And now currently we have this most commonly studied 3D printer that is from Cellink, that is called BioCellX. And you see here, this is in 1984. You see how it looks like. And you see, to get modernized, it took nearly 38 years of transition process. And why 3D printing is so fascinating? What is the reason? The reason is that there is an increase in use of 3D printed products in pharmaceuticals and cosmetics industry. And because of its beneficial properties, there is a rise in public and private funding to support all these activities. And you see here, these are the key players in 3D printing marketing. Most of them are in the US. So Bico basically selling, that is in USA. 3D Systems USA, Call Plant Israel, Envision Germany, RegenHU Switzerland, and in India we have this NABL, that is Next Big Innovation Lab, that talks about Trivima. Trivima is 3D printer. Okay, that is basically an indigenously developed 3D printer. Okay, Trivima. Okay, so this is NBL. They are located in Bangalore. That is called Next Big Innovations Lab. So they have this indigenous 3D printers that are developed in India. Okay, now you see that in the next 5-6 years, you see that the projected CGA CAR is around 20.8%, which means that from 1,284 millions, they're going to increase up to 3,304 million revenue. So this is what is projected in the next 5 years. And you see that the market is fragmented into many players, but most of the time it is from the Northern America, that is US, Canada, and etc. And you can use them for all of these applications. So for organ regeneration, bone regeneration, drug development, skin engineering, and medical devices. And the materials in terms of materials, they are fragmented or segmented as living cells, hydrogels, extracellular matrix, and other materials. So there is some difference between 3D printing and 3D bioprinting. So what is the difference is that when you print only the material, it is called 3D printing. When you print the material along with the cells, it is called 3D bioprinting. That is why the term bio is introduced. Bio is basically indicating the cells that you put inside your material and you print them. So what materials I can print? You can print n number of materials. So this is basically the natural sources: gelatin, collagen, chitosan, cellulose, decellularized organ, HAP, that is hydroxyapatite. So I mentioned earlier, I'll talk about this decellularized organ. So what is the decellularized organ? It's basically the organs that you take from the cadavers. You decellularize, as the term decellularize indicates, you remove the cells out from the tissues, you sterilize them, and you repopulate them with your mammalian cells. So the thing is that these decellularized organs are best suited for in vitro studies because that they retain the ECM component within them. Okay, so when the eSIM components is retained, which is basically mimicking the extracellular matrix, the cells will have more profound conditions to grow and expand and differentiate. So that is the reason why decellularized organs are most preferred in your cell culture applications. So if I have problems with all of these materials, I can also go for synthetic materials, that is either metals, synthetic polymers, carbon fibers, or graphite. So the concept is that 3D printers or bioprinters will basically extrude a specific substance from the machine to build a predefined structure. That is the concept of 3D printing. And in terms of extrusion bioprinting, we need something called as bio ink. So what is this bio ink? Bio ink are basically cell laden, you call them as human cells or mammalian cells, that involves the use of scaffolds or cross-linking agents. Sorry, involves the use of scaffolds or cross-linking agents to ensure that the constructs are properly developed to mimic the target tissue in terms of structure and function. So the process of 3D printing is that the first one, you have to go for data acquisition, which means that by means of some kind of some kind of imaging technology, either X-ray or CT, you take the blueprint of that particular organ. Then by means of modeling, SolidWorks, either by CAD or CAM, that is called as computer aided design or computer aided manufacturing, you make a blueprint. Okay, and this blueprint will be fed into your 3D printer, and using this blueprint, your 3D printer will print this particular structure. So for printing, you need bio ink. So bio ink is basically the cells, growth factors, and the scaffold. Here, the simple example is your hydrogen. And when you have put in the bio ink into the material, there are some parameters that you have to adjust. That is basically the bioprinting parameters. That is basically your resolution of printing, printed calibration, the physiological temperature and pH. After you have printed the material, this is something called post printing, where you try to functionalize the printed structures, and you can do them either by doing some kind of cross-linking or putting in them in an incubator or a bioreactor that will allow the cell growth and viability and proliferation and etc. And after you do the post printing, post bioprinting, you have to go for an application. So this application, it can be in your lab, that is called in vitro testing, or you can do some kind of disease modeling, or you can also go for in vivo implantation. So now let's discuss in detail what is this bio ink. Excuse me. Bio ink is basically the living cells and biomaterials. Two things are there. One is a cell, another one is a biomaterials. So the combination of cells along with the biomaterials are put together that basically mimics the extracellular matrix environment, which can allow cell support, which can allow cell addition, proliferation, and differentiation after printing process. So basically you have the cells, polymers, and cross-linker. And this is basically called as the triads of bioink. And this polymer can be either a single polymer, that is called single component bioink. If there are more than one polymers, you call them as multi-component polymers. And to make these polymers link with each other, then you need a cross-linker. So the properties of an ideal biomaterial is that it should have three essential properties. One is something called the functionality, other one is printability, and other one is your biocompatibility. So what is all this? So functionality is basically it should have the physical support to enhance cell stimulation via growth factors and biomaterial modelization. Printability is that they should exhibit some properties that is called as the shear thinning properties. We'll talk about them in coming slides in detail. Then you need some kind of gelation kinetics, suitable cross-linking. So this all comes under rheology and viscosity. In terms of biocompatibility, again, they have to be non-immunogenic, not eliciting any kind of immune response. They should have a controlled degradation rate, and the products of the degradation should not be toxic. And you see here, this is under gelation. Under gelation, the typical bioink should form a drop first. So this is okay. And after forming a drop, under proper gelation, there should be a continuous filament formation. Only if there is a continuous filament formation, you'll get this grid-like pattern. If it is over gelled, you see here, this is not a continuous pattern. It is not a filament. It is getting disturbed. So proper gelation is an essential characteristics that will allow the proper printability of your material. So again, gelation, viscosity, the nozzle gauge, shear stress, fabrication time, network properties are essential for your ideal bioink. So what materials can be used for bioink? You can also go for synthetic materials, natural materials, or the composite, where you have the two, natural with synthetic, or natural or synthetic composite. And also you can also go for decellularized matrix. Here, this is a typical example. This is a printing of gelatin, a human here, which is printed with gelatin. This is gelma, which is basically gelatin methacrylate. This is silicone, nanocellulose, and this is a sacrificial material which will allow the organ to form, and the sacrificial material can be removed. So when you talk about the printing, two parameters are very essential. One is a material parameter, and what is a cross-linking parameter. So for example, here let's take that you're taking sodium alginate, which is used for bio ink preparation. The molecular weight of sodium alginate is very important. The number of units in the sodium alginate is very important because this determines the viscosity and concentration. And in the 3D printing system, how you're going to cross-link the material. So what type of cross-linkers you're going to use? Again, the concentration and the volume is very, very important. So the different forms of bioink, you have matrix bioink, curing bioink, sacrificial bioink, and support bioink. So what are these? So matrix is basically, you have a matrix. In this matrix, you have the cells that are attached. So most common example is gelatin, alginate, or sometimes you can use a scaffold-free slurries. Curing is basically, you have a chemical cross-linker or a photo initiator, which will improve the gelation of the bioinks. Here, a typical example is Irga cure, or sometimes you can also use calcium chloride. Sacrificial is a material that will be removed after printing. So they will support the system to allow the printing process. But when the printing is completed, you can remove them. So it is a temporary support which will allow the vascular network development. Simple example is your hyaluronic acid and agarose. The last one is something called as your support, the materials with the sufficient mechanical properties. So they will provide good mechanical properties. Here, most of the time, we use all the synthetic materials, that is PCl, that is polycaprolactone, PLA, polylactic acid, or their PLA, PGA, PLA combination, that is PLGA. So when you talk about 3D printers, there are multiple types of 3D printers. So there is something called inkjet bioprinters, which will give you only the droplets. There is something called extrusion bioprinting, which will give you as a filament. Then there is something that is called laser assisted bioprinting, or there is something called SLA, that is stereolithography. But here we will focus only on this extrusion bioprinting. And all of these printing technologies have their own limitations and advantages. For example, if you take here in terms of resolution, the inkjet bioprinter has high resolution followed by laser and stereolithography. In terms of print speed, the fast can be done by inkjet, and again fast can be done from SLA. Extrusion is very slow and laser is medium. And in terms of cost, this is low, this is somewhere medium. So most of the time for my studies, I use extrusion. This is very, very costly, and SLA is very, very low. So here in the coming slides, we'll focus only on extrusion-based bioprinting. So what is this extrusion-based bioprinting? Under this extrusion-based bioprinting, you have three different concepts. One is pneumatic dispension, other one is piston-driven dispension, and the third one is screw-driven dispension. So what are these processes? So pneumatic is basically you have the air. The air will push the material. So based upon the air pressure, you get the printing, that is called as your pneumatic dispension under pressure. And pistol-driven dispensing is that you have a pistol that will push down your materials and you print it. Screw is basically you have a screw that you rotate it. Okay, so all of them are based upon a particular force that acts on the material to pull the material out from the syringe. So the simple analogy is basically you're squeezing a ketchup out of your bottle. So I'll show you here. Squeezing a ketchup out of your bottle. Why is that amazing? Okay, I'll show that later. Okay. So when you want to talk about the optimization of a bioprinting process, there are two terms that needs to be understood. One is something called as a pressure, another one is your printing speed. So what is this pressure? Pressure and printing speed, I'll give you an example, the trade-off between high pressure and low pressure. You see here, this is a low pressure printing, you have a proper structure. When you increase the pressure, you see that you experience a discontinuity in the filament formation. So this is not desired. So what we want is a low pressure printing, which gives you a uniform structure. Then the speed, the printing speed and the filament length. You see here, this is one, you have a proper filament. And when you increase the speed, what happens? You see here, when you increase the speed, you see here, increase the speed, the filament size is getting reduced. And above this particular speed of 6 mm per second, again you exhibit a discontinuity. So this optimization is very, very important. So here we have optimized saying that between 1 to 5, we can get a good print speed. with a proper filament structure or a filament width. But when you exceed the 6 millimeter, 5 millimeter per second, we have a discontinuity in the filament formation. So this trade-off between the pressure and the printing speed is very, very essential. So this optimization of these printing parameters are essential to get a good construct with the proper shape fidelity. So this is from our experience. You see that we have a different concentration. These are multi-components. One is cellulose nanofibers and other one is alginate. You see we have in different compositions, and you can see that this composition, that is 80:20, we were able to get the typical grid-like pattern. And we printed this material using Cell-Link BioInk, Cell-Link BioX printer, and we had the optimized pressure that is somewhere between 20 to 60 kPa, and the nozzle temperature was 25. The bed temperature, printing bed temperature is 37 degrees, and the speed you see here, we optimize the speed between 5 to 15 per second. So if we exceeded this printing speed, what happened? We will exhibit this kind of discontinuity. You see here, there is no grid formation. There is a total discontinuity here. So the nozzle diameter was 22 gauge, and interior fill percentage was 5%, and the grid size was basically 20 to 20 to 1 mm. And for cross-linking this, we had this 90 millimolar of calcium chloride for 20 minutes. So this is the printed structure, and post printing, you see that we are able to handle this material. So this is using a finger and this is using your forceps. So again, the second optimization is, first one we saw about the pressure and the print speed. The second optimization is a cross-linking mechanism. So what type of cross-linkers that you use to bind all your proteins together and you exhibit a three-dimensional construct. So cross-linking basically talks about the printability of the material. Here we have two concepts, shear thinning and photosensitivity. So when you have the cross-linking, you can go either for physical cross-linking, chemical cross-linking, or enzymatic cross-linking. And in physical cross-linking, you have the non-covalent interaction. In chemical cross-linking, you have the covalent interaction. And these are, again, printer-specific. Physical cross-linking only possible in your extrusion-based bioprinter. Chemical cross-linking, again, it is possible only in light dynamic processing, light-based bioprinters or extrusion bioprinters. Again, in enzymatic, again, either the light-based or the extrusion-based bioprinter. So the different cross-linking mechanism, there should be a proper formulation. So that is why a compromise between the degree of cross-linking and printability is very important, because the lower cross-linking density will enable faster bioing flow, but the shape will not be good. That is why it is called as a poor shape fidelity. On the other side, higher cross-linking degree may lead to stiffer gels. It will become stiff with a better stability, but it will not allow good printability. So the compromise between the proper cross-linking degree and printability is very, very essential to produce a proper structure that can flow out of this syringe. So how do I assess the printability and shape fidelity? So initial screening can be done where you have the first drop formation or the filament formation, or also you can try to understand the layer stacking or merging. In terms of rheology, you can understand the shear thinning property. We'll talk about the shear thinning property. And there is an important property that is called as your thixotrophic property. So that is that the property of the gels or the fluids that are normal, that is basically normally thick, that is basically viscous, but they become thin when they are stressed. So this is something similar to your biological fluids in your body. So all your biological fluids in your body exhibit this viscoelastic property that is called as your thixotrophic behavior, or you call them as simply as a shear thinning behavior, which means that under stress condition, the viscosity decreases, and when the stress conditions are removed, they go back to the original form. So that is very important. So this is a simple example, squeezing of ketchup out of the bottle. You see here, you have a ketchup, and ketchup is a simple example of your thixotrophic property. It does not flow. Even though you tilt the bottle, it does not flow. But when you apply some kind of force or something, the sauce is coming out of the bottle. So this is a basic thixotrophic behavior of your of a typical sauce. The same thing happens in your body fluids. So when you have this thixotrophic behavior, we also talk about something called as your storage modulus and your loss modulus. Again, these are all rheological properties which determines how your material is able to exhibit this viscoelastic property with response to your shear thinning behavior. So storage modulus is basically the ability of the material to store energy elastically. And loss modulus is nothing but the ratio of the viscous and the elastic phase that is able to dissipate the heat during the stress conditions. So this is the difference in shape fidelity of the material. You see here, this is the SDL file. This is the printed structures. And this is the free handling of the material post printing. You can see they are handled. And basically, whenever you try to print something new, you first print a grid-like pattern. When you're able to achieve this grid-like pattern, then you go for complex structures. So you might be wondering why all the structures are in grid-like pattern. The reason is that first you print a grid-like pattern with four cells. When you achieve this, you increase the patterns. So you have this type of structure, and you should be able to stack them, going them in the y-axis. You should be able to stack them. When you achieve all of these without any collapse of the structure, then you can try to produce 3D solid organs with more complex geometries. And here you see that this is basically a cellulose nanofibers. So this is 1% cellulose and this is 5% cellulose. So again, the concentration of cellulose is very important. When you increase the concentration of cellulose, you see you're able to get a proper structure. This is basically something called as a dog bone shape structure that is used for mechanical testing. And this is a cylindrical structure. You see that this is the SDL file which was fed into the machine. When you increase the concentration of CNF, that is cellulose nanofibers, from 1 to 5, in the concentration of when the concentration is 5%, you can see that it is able to produce a proper structure. So increase in shape fidelity with increase in polymer concentration. So to wind up this, there is something called as your biofabrication window. So what is this biofabrication window? So the biofabrication window says that when you have an ideal cell culture, there is a poor shape fidelity. But when you have the ideal bioink, there is a poor cell culture. So that when you go on this side with complexity of the material, there is a poor cell culture. So you can build strong tissues here. You see here, you start something like this and you go. Increase in complexity, you exhibit structures with printability and shape fidelity. But at the same time, on the other side, ideal cell culture with poor shape fidelity. So the reason is that now we are somewhere here, the traditional biofabrication window. In future, we should advance to this particular stage where we should fabricate bioinks which are able to achieve both the complexity as well as the biocompatibility without any compromise. So the future prospects of 3D printing, where ideal bioinks are fabricated, should be tailored with both the biological as well as the mechanical properties. So these are some of the recent examples of 3D bioprinting. And this is something called a skin 3D bioprinting that is using a customized handheld printers to heal incision or excision wound. Because you see here, this is a handheld printer, and if you have a wound here, you simply put the machine on this place, and the machine will have the ink, and you simply print it. So this is something called as your in situ printing of the biomaterials onto the defect. And this was evaluated in a pig model. You can see here. Then for bone 3D printing, again, this is a cranial defect in a rabbit model. You can see that these are the defects in the brain, and you can 3D print structures that are basically having the features that are similar to the natural bone, which means that it will have 70% of hydroxyapatite and 30% of collagen. So this can be implanted and you can study them. So this is basically PCL. Then this is in terms of cartilage, where in terms of cartilage, we try to understand the osteochondral defects. So you see here, this is a scaffold. This is a native, and this is with the help of TGF beta. That is the growth factor here. And again here, the stacked layers, which are trying to mimic the osteochondral layers, because the osteochondral layers have three different layers. The first layer is the cartilage layer, calcified layer, and the subchondral bone. And the subchondral bone only has the collagen, but here you have this hyaluronic acid, and along this here you have the cells. So trying to mimic the three different zones of the cartilage. Vascular, trying to grow vascular vessels. You can see here they are printing vascular grafts with the dipyridamole. This is another example trying to achieve the dimensions in terms of outer layer and inner layer. You see these structures which are 3D printed, and you can see here, you see the height of this material. They are not collapsing, which means that the shape fidelity of the material is being retained. And finally, this is our experience with 3D printing. So we also do 3D printing. This is a basic grid-like pattern that we developed. And this material is basically cellulose nanofibers and alginate. And when you do a SEM, you see here, this is a SEM morphology of the 3D printed structure. And you can see there are a lot of pores here. So these pores are very, very essential for the cell penetration activities. And this is a live death staining. And this one is your Hausches staining. And again, they are compactable. This is an MTT assay. So with this grid-like pattern, we wanted to print complex structures. So this is basically the composition of uh, alginate and your CNF with different compositions. So these are all the yellow color ones are poor shape fidelity, and these are the printable regions, and this one is non-printable. And you can see here, this is basically a vascular tree, and this is a meniscus which is 16 bar 16, and this is a hollow meniscus which is again 8 bar 8 layers, and this is a human here. You see that we were able to print this complex structure human here, so perfectly printed with 27 bar 27 layers. Then I'll just have this. I don't know if I can play this. So you see here we are printing the human here. You see how it prints. So the material is extrudable from the syringe, and the material exhibits some kind of shape fidelity. That is why we are able to print layer over layer. If the material is not extrudable properly, and if there is no shape fidelity, it will collapse completely. Okay, and this is the same thing with the hollow meniscus. We're able to print structures. Okay, so it is a long video. I'm just pausing here. So what are these challenges in 3D bioprinting? So there are benefits, there are also challenges. The challenges is that, just a moment. The challenge is that we can fill the wide gaps between organ demand and availability, because we cannot depend upon organ donors for every time. So if there is shortage of organ, we need not depend upon the donors. We can develop tissue engineering replacement grafts that can be used instead of donors. Organ less likely to be rejected after implantation, and there is a more precise and quicker method for artificial organs. So what are the challenges? Are we able to really mimic the real organs? Yes, we are not able to mimic the real organs, because there are not only a single type of cells that develops an organ, there are multiple types that are playing a vital role in organ development. And developing and translating the complex tissue systems requires a very coordinated and synchronized systemic functions. That is very, very, very challenging to achieve. And we are also lacking in 3D models that can be used for software analysis for the tissues and organs. So in summary, any product that you have in tissue engineering, it should be scalable and saleable. And obviously, there is a hope as well as a hype in 3D bioprinting. So the question is whether 3D printing is a hype or a hope. I believe it is a hope. It is going to transform. It is basically an evolving technology. Though we started in 1984, now we are in 2025. A lot of things have happened. So initially we were in this TRL1. Now we have products at TRL5. So we are able to develop products that can be directly developed in the labs and can be directly go to the human applications. So definitely it is a hope. Way to go. More evolving technologies or concepts are yet to come. So in a nutshell, the choice of the 3D printer, choice of a bioink depends upon what type of tissue that you want to repair. And bioprinting process parameters greatly influence the printing resolution and the shape fidelity. So these two parameters are very, very important in controlling your printing resolution and shape fidelity. And post-processing, basically after you print, is essential in obtaining optimized implants in terms of shape, size, and contour. So I would like to acknowledge my funding agencies. So I have funding from National Institute of Technology, that is from Calicut, Tamil Nadu State Council, INSA, DBT, and SCRB. So these are my students with me who have worked with me, who are working with me, and these are my research collaborators. So with this, I end my session. Thank you for this opportunity, and the questions you have, I'll be able to answer them.